My name is John Papshak, and I work in operations at Biotia. I'll be taking you through a quick demonstration video of our COVID-DX software. At Biotia, our mission is to fight infectious diseases with genomics and artificial intelligence while building the world's leading microbial sequence database. The SARS-CoV-2 virus has caused a global pandemic of the 2019 novel coronavirus, or COVID-19. Real-time reverse transcription polymerase chain reaction-based methods are good at qualitatively detecting SARS-CoV-2. However, SARS-CoV-2 is mutating and evolving. These mutations may impact how well common testing methods can detect SARS-CoV-2, how quickly the virus spreads, how effective vaccines will be against the virus, and how an individual patient with the virus should be best treated. Detecting mutations requires novel technology, so we work together with Twist Bioscience to develop a new SARS-CoV-2 test. This technology includes hybrid capture-based targeting of viral RNA, next-generation sequencing, and proprietary bioinformatics and artificial intelligence analysis. We have also extended this software to work with the amplicon-based NGS ARTIC protocol. Using these technologies, we have developed the COVID-DX software. Our software characterizes SARS-CoV-2 from NGS data. Our user-friendly portal enables bulk ordering of the software. Our reports describe the presence or absence of SARS-CoV-2, lineage, genetic variants identified, along with literature and CDC insights into those variants, the virus's clade, the virus's phylogenetics, and performance details of the sequencing run. We also provide the Fasta files and a spreadsheet of the results for all the samples analyzed in a batch. In the next section, I will demo the COVID-DX software. Now I will demo how to use the Biotia portal in order to create a COVID-DX software service order. You must first sign into the portal with your sign-in credentials. If you do not have an account, you should click the Register button to create an account. You will then get an email notification saying that your account must be approved by an admin. Once approved, you will be able to sign in. You will receive a verification code to your email. Once you enter the code, you will be taken to the portal home screen. Once you sign in, you will see 3 tabs on the left-hand side, which are Home, Order Creation, and Order Lists. Order Lists will show you all of the previous orders you have associated with your account. Order Creation is where you can create a new service order. Click this tab and you'll be taken to Software Service Order page. You can find your name by typing into the search bar. You will be prompted to fill out a few fields and check some acknowledgements. You will first be asked to accept the Biotia portal terms of use by clicking on the slider. You can review the terms of use by clicking on the blue link here. Then you enter your product serial number in the appropriate space. For Twist customers, you will use the queue number emailed to you as part of your order for the serial number. For platform, you will choose between the 2 targeted next-generation sequencing methods, ARTIC and Twist. You will then be required to check the slider to acknowledge the payment of applicable charges for the service. Software credits are provided free with purchase of a Twist SARS-CoV-2 NGS assay, and other credits can be purchased by contacting Biotia. After that, you will acknowledge that the contents of the research use only report will be used solely for research purposes, and will not be used as the basis for any clinical diagnosis. You will then fill out the researcher data section, which includes your name, email address, and the institution you are associated with. After you complete these forms, you can select the Add Test button on the bottom right section of your browser. The test will take a moment to generate, and soon you will be able to scroll down to a new section called Test List, which will have 3 areas: Download Order Report files, Download Submission Form, and an area to drop your completed submission form files. You will click the Download Submission Form button, which will download an Excel submission file into your downloads. The next step is to fill out this form with your Fastq sample information and then upload the updated file into the Upload Submission Form section. We have also added the option to add your Fastq sample information directly from the portal through the Add Test Manually button on the right side of the test list section. You can see a more in-depth explanation of all of these fields in the next section where I go through the Excel entry demonstration. Now open the Excel spreadsheet from your downloads. At the top, there is a customer information section that is auto-populated from the information that you filled out in the previous step. Below that are columns where you fill out information about your samples. Some sections are required and some are optional. Column B is the customer sample ID, and it is required. You can enter your own sample ID in this section. If you have multiple Fastq files associated with a single sample, you will enter the customer sample ID in as many rows as you have Fastq files. For example, if you have 4 Fastq files for a sample, you will enter the same customer sample ID in the first 4 rows. I name my sample 6-10-21-GO-0250, which is based on my Fastq file. And since I have 4 Fastq files associated to my sample, I repeat the customer sample ID for 4 rows, as shown below. Column C is the requested test type and is also required. If you click on one of the entry boxes, you will be presented with a drop-down menu of options to select your test. The options are COVID-DX for research use only, COVID-DX for emergency use authorization, which is the FDA version of COVID-DX, and Biotia DX. For this demo, I'll be selecting COVID-DX for research use only. Column D asks for sample type. The drop-down menu gives the options negative control, negative extraction control, positive twist RNA template control, internal control, and test sample. We recommend that you use the controls in the protocol, although they are not required if the test runs as research use only. All of these controls listed are required for the SARS-CoV-2 NGS assay authorized by the FDA. For this test, I selected test sample for all 4. Column E asks to input serial number if it is different from the top. In this case, it is not different, so we can skip this column. Column F is the most important as it asks for the Fastq file names. At the moment, we only accept the Fastq file names in the fastq.gz format, which is the compressed form. This makes it quicker and easier for upload. You can see the format below of my Fastq file names. I copied and pasted the exact file names from the file and pasted it into the Excel form. It is important that your fastq.gz file names are correct, because when you upload the actual Fastq files in the next step, if there is a mismatch between the file names on the spreadsheet and the actual file names, the program will return an error. Next is column G, which asks for checksum entry. Checksum, or SHA-256 checksum, is a short string that lets us verify that a file uploaded properly. If any of the data in an uploaded file is changed or corrupted from the original file, the new checksum will be different. The checksum is not required in the submission form for the research use only version of the software. However, it is required for the FDA emergency use authorization version of the software. If you are curious how to find the checksum for a file, please refer to the FAQ section on the Biotia website. Column H asks for original specimen type. However, this section is also optional. This section also has a drop-down menu with a variety of specimen types that you may select. Columns I, J, and K have various dates relating to the samples, which are also optional. And the following columns, L, M, N, O, and P, have additional optional demographic data. Once the form is completed and all the information is correct, save the Excel sheet and upload to the Upload Submission Form section. You may either drag and drop or select it from your files and hit Upload. Once your submission form is uploaded, you will see that the table below is populated with the contents of the Excel file. Next, you can drag and drop your fastq.gz files into the appropriate spot. For quicker upload of large files or many files, you may instead do an API upload. After both the submission Excel form and fastq.gz files are uploaded, you can click the Submit button. Now the order is sent to the Biotia pipeline for processing. It will take anywhere from minutes up to a few hours for the analysis to be completed. You will receive an email when the results are ready for viewing. You will also be able to tell that the order is complete by looking to see if this icon is colored in. When the order is complete, you will see that this download icon went from gray to orange. If you click this icon, you will be able to download 2 files for each sample, the report and a Fasta file for further downstream analysis if desired. The SARS-CoV-2 consensus Fasta file we produce can easily be submitted to NCBI and GISAID. You may also download a summary Excel for each batch of samples you have submitted for the analysis. To download the batch summary, click into the order and click on the red button Download Summary. The report has many details from your sample's analysis. The report summary shows whether COVID-19 was detected, as well as variant lineage and clade. We also show any genetic variants that were detected, variant curations, and lineage curations. Additionally, we include a chart of variants and coverage across the SARS-CoV-2 genome, a phylogenetic tree which shows where the sample fell evolutionarily, assay performance metrics, and methods and limitations. You may also view the second file, which is a Fasta file. Specifically, this is the Fasta file of the genome of the particular strain of SARS-CoV-2 that was identified in the patient. Thank you for watching our presentation. Please contact sales@Biotia for more information.